How's everybody doing? I brought note cards. Is that good news? You guys want to come up and get them? So here's the deal on note cards. So they're big, all right? You can write on both sides. Everything you put on the note card has to be handwritten. You can't tape a figure on there. You can't print a note card, that is with a printer or something. You have to put everything on there by hand. You need to put your name on there, and you need to turn it in when you are done. And everybody has to turn in a note card. So if you don't turn in a note card with your final that you got from me, you're going to lose about 10 points. Okay? So, everybody understand that? So for those of people out in TV land, except for the e-campus, that's different, but everybody else out in TV land who sees that knows that you need to get the note card from me. So I'm not going to bring them back on Friday, okay? And I'm not going to be in town on Monday, and I'm not going to be in town on Tuesday when the exam is done. So if you want to get a note card from me, you will need to get it no later than 5 p.m. on Friday. Everybody understand that? You guys understand that because you're here, but TV land people need to make sure they understand that too. So it might be a good idea to watch the video before Friday. Yes, ma'am? Good question. Review session is going to be problematic. I haven't decided. I will try to do a review session on Saturday afternoon. That's my tentative plan. But it kind of depends on my schedule, and I haven't committed to doing that yet. I need to look at my schedule. If I cannot do a review session on Saturday, what I will likely do is post last year's review session that I did so that you'll have something to watch for a review session. Does that make sense? Yes, ma'am? No, I don't post old finals, and the reason I don't do that is I don't like to encourage people studying old exams. I think that's a bad idea, so I'd rather have you studying the content. So no, I won't post that. Okay? And in my experience, getting you to understand, because the format's the same. I'm not going to change the format. Getting you to understand the content is the most important thing. And I find that when students focus on exams, I don't do that to be mean, they really focus very hard on what's there, but the exam's going to be different, and they won't focus on the things that we've covered in the class. So I really do try to make the coverage on the exam roughly proportional to what you've had in the class. So the new material that will be on the exam will be proportional amount of the exam, roughly 25 to 30%. Was that your question back here? It's the most common question, yeah. Everybody understand? Okay, so come up and get a note card from me. You have to get it from me. Don't get one for your friends. You're just getting one note card, and this is it. So, there you be. You bet. Okay. Yep. Okay. Oop. Maybe. Sure. You bet. You bet. Sure. Sure. You bet. Study hard. Thank you. There you go. There you go. There you go. And there you go. All right? Okay. Oh, and there you go. Okay. Last but not least. All righty. Now, we are on our last topic. And our last topic is very relevant for nutrition. It's very relevant for actually all areas of biochemistry because it relates to nitrogen metabolism. And so nitrogen metabolism includes things like how amino acids get made, how amino acids get broken down, how nucleotides get made, how nucleotides get broken down, and how the body manages nitrogen. It's a lot of stuff, right? Well, I'm going to give you the highlights of all of those things. And what are we doing there? Okay. The highlights of all of those things, and try to cue you in on the relevant stuff, particularly for the exam. Okay? Now, nitrogen metabolism is interesting, okay? Because nitrogen is not something that we pull out of the atmosphere. We breathe it, but we can't do anything with it. Okay? So the nitrogen that we have in our body in the form of amino acids and so forth is all there as a result of things that we've eaten or drunk in some cases. Okay? So we're not getting any nitrogen from the atmosphere. In fact, getting nitrogen out of the atmosphere, as I will show you in just a little bit, is a real problem biologically. It's a real problem biologically, getting nitrogen out of the atmosphere. This is accomplished, for those of you who are botany plant pathology majors, by what are called nitrogen-fixing bacteria. And nitrogen-fixing bacteria are what we owe, essentially all of the nitrogen in amino acids, in nucleotides, and so forth. We owe that debt to them. They're the ones who pull it out of the atmosphere. And it's energetically very costly for them to do so. And consequently, that's why I say we want to be careful thinking about what we do to the soil, what we do to the water, and so forth, because these organisms make it possible for us to have nitrogen. And we have to have nitrogen because we have to have amino acids. We have to have nucleotides. We have to have other things. Okay. Well, this schematic sort of shows you the various ways in which nitrogen comes in to basically the biosphere. It really starts with nitrogen-fixing bacteria, and there's some nitrifying bacteria and other nitrifying bacteria that do that. But they're the ones who are pulling this nitrogen out. Many of you know about legumes, and legumes are plants that contain bacteria that are able to assimilate this nitrogen and provide it to the plant. So in the case of beans, for example, beans are legumes, and you can see a little bean plant there. They have bacteria in their roots that fix nitrogen for them and allow them to use nitrogen from the atmosphere. Okay? That's really important, again, because without that, then the plant would have to find some other way of getting the nitrogen. Okay? Now, not all plants are legumes, obviously. Okay. Nitrogen makes it into bacteria in several ways, but the most common way I'm showing you right here is through nitrifying bacteria. And nitrifying bacteria, okay, nitrogen-fixing bacteria, I should say, nitrogen-fixing bacteria start with molecular nitrogen, which you can see right there. Okay? They add a total of 16 electrons. They add a total of 8 protons, and they make 2 molecules of ammonium ion, which is NH4+. Okay? That's very, very energetically costly, as we shall see. Okay? In fact, you can see right here, 16 ATPs to make this process happen. Okay? So it's very, very expensive energetically to fix a single molecule of nitrogen into 2 ions of ammonium. Okay? Well, nitrogen turns out to be not only important, but there's a yin and yang to it. Nitrogen in certain forms is also toxic. Okay? So ammonium ion readily breaks down into ammonia, and ammonia is toxic. It's problematic for you. So any living organism has to spend some time balancing the amount of nitrogen that it has. Okay? We're going to see some examples of that, particularly as I show you the urea cycle in a little bit. But organisms have to have ways of bringing nitrogen in, and ways of balancing their nitrogen, and ways of getting rid of excess nitrogen. Okay? In our bodies, our exit mechanism for nitrogen is the formation of a compound called urea. And urea is the thing that gives your urine the smell. Okay? nitrogen compounds frequently smell. Okay? So that's what's happening with that. Because nitrogen is difficult to get in, nitrogen is precious to cells in addition to being toxic to cells. Okay? So once we've had nitrogen in a reduced form, and we're thinking about in an ammonium or ammonia type form, okay, then that nitrogen is carefully distributed through metabolism according to need. And there are some very important players in this process, as we shall see. The two most important for our purpose are right here. Glutamate, which is the same as glutamic acid, and glutamine. Now, you probably don't remember your structures, but I will tell you the difference between these two structures. The difference between a glutamate and a glutamine is an amine group. Okay? So this guy has an amine group on its R group carboxyl, making it I can't think of the word. I'll come up with the word in a minute. But it has, on its R group carboxyl, instead of the double bonded oxygen, it has an amine down there. Okay? All right? And the glutamate has a single alpha amine located on it. Okay? Carboxamide, that's what it was. It's called carboxamide. All right? Now, we can see the entry here of ammonium ion. Yes? Glutamate only has one amine nitrogen. Glutamate has one amine nitrogen. Glutamine has two. Okay? And so you can see that if we go from glutamate at the top to glutamine at the bottom, we have to add an amine, and the amine is coming in the form of that ammonium ion right there. So ammonium ion, when we add it to glutamate, we get glutamine. That, too, is energetically costly. We have to spend an ATP to get that in. Okay? And this figure is pretty complicated. All right? The figure is pretty complicated. This glutamine can donate nitrogen to help form any of these compounds here. Serine, carbamyl phosphate, glucosamine 6-phosphate, tryptophan, histidine, CTP. Any of these guys can get a nitrogen from glutamine. So glutamine's got two. It's got one to give. One is in the R group, and it passes off that nitrogen readily. Okay? So glutamine is a distributor of nitrogen. It's a carrier of nitrogen, and it's carrying it in the form of an amine. When I say an amine, I'm talking about an NH2 group, like on an amino acid. Now, this figure's complicated because we look at some of the products that come off of glutamine, and we see these little dotted lines going up here, and they go to these little red circles. Well, you know from driving on American roads that that means no. So all of these compounds are inhibitors of the formation of glutamine. Now, I'm not going to ask you to memorize this chart because that's not my reason for showing it to you. But I show it to you to give you an idea of the complexity and the balance that cells have to exhibit in terms of the movement of nitrogen between molecules in the cell. Okay? There's a fair amount of balance that has to happen. So the cell doesn't want to make too much glutamine. All right? And we'll see what the cell does if it has too much of the ammonium ion later. But if it's needing these compounds, then it will make glutamine, and then the glutamine will pass off the nitrogen to the compounds as needed. Does that make sense? Okay. This figure shows how it is that cells can make glutamate and glutamine. Now you can see the structures that I'm talking about. Okay? The glutamate can be made from alpha-ketoglutarate, which of course you know is an intermediate in the citric acid cycle. And when we talked about the citric acid cycle, I told you that there were enzymes that could readily convert alpha-ketoglutarate into glutamic acid. That's what you see happening right here. Okay? And here's glutamate being formed. Okay? So we see this double-bonded oxygen that was on the alpha-keto group has been replaced now by an amine group. Glutamate, which was the end product of this reaction, is the starting point for this reaction over here. And we see that glutamate can absorb another ammonium ion and become glutamine, as you can see over here. And I earlier said that replace the double bond oxygen, it replaces the OH. I'm sorry for the mistake. Okay. Here are the two amines that are found in glutamine. One here, that's the alpha amine, and one down here, that's the R group amine. Okay. Now, it turns out that the movement of nitrogen between amino acids is actually more interesting than what you saw there. Okay? There, and I thought I had this on here. Here we go, right here. Okay? It turns out that amines in the alpha position for several amino acids are readily transferred to other compounds. And I've also referred to this earlier in the term. These are reactions called transaminations. This is a very important set of reactions I'm showing you on the screen here. Transaminations are one way to move amines between amino acids and other things. So in the last slide I showed you how to make glutamate and how to make glutamine. Now I'm showing you how I can take glutamate, for example, and transfer its nitrogen, its alpha amine group, to another molecule. This process is called transamination. How does it work? Well, we see here on the left, there's glutamate, glutamic acid. There's its alpha amine shown in the square. And here's an alpha-keto acid. This could be, for example, pyruvate acid, pyruvate. So the R group there would be a CH3. Okay? So if I had pyruvate, the R group would be a CH3. And there's an enzyme called a transaminase that will catalyze this reaction. In fact, we're going to call all the enzymes transaminases. All the ones that categorize this type of a reaction. You'll see there's many. So you'll notice this says alpha-keto acid. This could be alpha-ketoglutarate. This could be aspartic acid. Okay? Not aspartic acid, I'm sorry. Oxaloacetate, which makes aspartic acid. Okay? Anyway, let's follow this reaction. So here's the alpha amine group. The alpha amine is moving from the alpha position onto the oxygen position of the alpha-keto acid. And the oxygen that's on the alpha-keto acid is conversely moving back to the amino acid. So what have I done? I've swapped this with this. That results in the production of alpha-ketoglutarate and an alpha amino acid. So if this were pyruvate, this would be alanine. Okay? So we make glutamate, and now we're moving that amine from glutamate to another alpha-keto acid to make an amino acid. Okay? So this reaction can happen for a variety of things. Here's an example. Glutamic acid plus oxaloacetate. It's an alpha-keto acid because there's the alpha carbon, there's the keto. Okay? We swap the oxygen with the amine, and we get alpha-ketoglutarate, and we get aspartic acid. Don't worry about the enzyme name here. We're going to call all of these enzymes transaminases. Okay? So the enzyme is transaminase. So a transaminase catalyzes the swap of an alpha-keto oxygen and an alpha amine group. That's what they catalyze. Questions about that? Seeing the movement of nitrogen between different molecules. Now this turns out to be a very important pathway for the movement of a lot of alpha amine portions of molecules. All right. Now we move to more general things. I've shown you a specific example, and you could imagine this would happen for a variety of alpha-keto acids. Okay? It turns out when we study amino acid metabolism, that amino acid metabolism, we see that we've got 20 amino acids that our body needs and uses. Some of those amino acids we synthesize. Some of those amino acids we have to have in our diet. And as nutrition majors and other majors, I trust that you know that whenever we put the word essential in front of something, it means it must be in our diet. We talked about essential fatty acids. Those were fatty acids that had to be in our diet. When we talk about essential amino acids, these are amino acids that have to be in our diet. We're studying in each case here groups of amino acids that have common precursors. Things that have common precursors. Alpha-ketoglutarate is a precursor of glutamate. Glutamate is a precursor of these three amino acids. Okay? Now I'm going to show you all the various families. I'm not going to ask you to memorize the families. I want you to recognize that there are families, and it wouldn't hurt for you to know what the families are, but the individual components I'm not going to ask you to know. Okay? Now, you're going to see up here that some of these I'm going to show non-essential amino acids, meaning things that we can make. In other cases, you're going to see essential amino acids up here that we can't make. So how am I showing you pathways? Remember, the pathways are for certain organisms. So we can eat, for example, a mushroom that can make histidine where maybe we can't make it, but the mushroom has a pathway for doing that. So the pathway I'm showing you is how a cell that can make that would make it. Does that make sense? So I don't want you to think that I'm showing you only human pathways, but rather pathways that a cell who can make that amino acid can make. All right. So alpha-ketoglutarate is one family, and you see there's several amino acids that are made in that family. Aspartic acid has a family of its own. Okay? Oxaloacetate to aspartate. Does anybody remember the difference between aspartate and asparagine? Does anybody know the difference? Since I didn't actually memorize the structures, you probably don't, but I'll tell you. Asparagine is the equivalent of glutamine for glutamic acid. Glutamine had 2 amine groups. Glutamate had 1 amine group. Aspartic acid has 1 amine group, and asparagine has 2 amine groups. Okay? So it's very much equivalent to the glutamic acid and the glutamine pairing, the aspartic acid, Gesundheit, and the asparagine molecule. Okay, well, this family, as you can see, has several things in it, and it starts with oxaloacetate. So here's a citric acid cycle intermediate that can result in the production of 1, 2, 3, 4, 5 amino acids. Here's a serine family. And notice now the serine family has as its precursor 3-phosphoglycerate. That molecule is popping up left and right. You saw it in glycolysis. You saw it in gluconeogenesis. You saw it yesterday in the Calvin cycle. And now you're seeing it as an essential part of the family that results in the production of serine and these other amino acids. Okay? Very, very important molecule. Here's the pyruvate family. Pyruvate, of course, is the end product of glycolysis. We see that pyruvate can readily be turned into alanine. I've already shown you that reaction, actually. Can also be converted into valine and leucine. And I'm not expecting you're going to memorize which ones are in which family. Now, part of my reason for showing you this is to sort of remind you of the centrality of many of these molecules in the overall metabolic scheme. Oxaloacetate was a molecule that was found in gluconeogenesis. It was a molecule that was found in the citric acid cycle. It was a molecule that we saw yesterday was found in C4 plants. Okay? And I tell you that to remind you that metabolic pathways, and this is something students, I want you to take away after you've had this class. Okay? What I want you to take away after you've had this class is that metabolic pathways are man-made inventions. We draw this square around here and we say this is glycolysis. And students tend to think of that is separated from everything else. But you're now seeing that some of these molecules that are in glycolysis are found in amino acid metabolism. You've seen them in the Calvin cycle. So that what's really happening inside of cells is more of a mixing and matching of these molecules as needs and demands change on those cells. So these man-made designations that we have of glycolysis or the citric acid cycle doesn't mean they're in isolation. They're actually part of that bigger picture of what's happening inside of the cell. Okay, here's the aromatic family. Aromatic amino acids, of course, include phenylalanine, they include tyrosine, and they include tryptophan. You can see all of those there. You can see a couple ways of making tyrosine. Okay. And look at the precursors here. Phosphoenol pyruvate, that's PEP from glycolysis. That's PEP from C4 plant metabolism. Okay. Erythrose 4-phosphate, where did you see that? Well, you saw that in the pentose phosphate pathway, and you saw that yesterday in the Calvin cycle as well. You probably didn't notice it because I didn't point it out to you, but again, these things are found in a variety of metabolic pathways. That bug is odd. Where's the bug at? It must be flying around up here, I guess. Okay. See him? Okay. The histidine family is the easiest one to memorize. It only has histidine in it. Okay. Histidine is an odd amino acid, and it's made from ribose 5-phosphate. Ribose 5-phosphate, of course, is the precursor of ribose, which is used in nucleotide metabolism. Okay. This complicated figure sort of shows you schematically what the last few figures were showing you with individual diagrams. And again, I'm not asking you to come back and regurgitate this figure to me. That's not the point of it. Rather, the point is to show you the relationships of a cycle like the citric acid cycle to all of these different amino acids. Okay? It's broken up into catabolic reactions and anabolic reactions. What's the difference between a catabolic reaction and an anabolic reaction? What does catabolic mean? Break down, right? Anabolic means make, right? So we see that many amino acids break down into intermediates in the citric acid cycle. So when we break down certain amino acids, we can get energy. People on high-protein diets, low-carb diets, are getting energy through these kinds of reactions. Okay? Many amino acids break down into acetyl-CoA. And acetyl-CoA can, of course, feed either of these cycles, breakdown or synthesis. Right? And last, we see over here that these amino acids, as you see here, can take intermediates from the citric acid cycle away and make amino acids. So, for example, alpha-ketoglutarate family included glutamic acid, glutamine, proline, and arginine. I think it was actually called the glutamate family, but the point is that it's taking an intermediate from the citric acid cycle to make this happen. So if the citric acid cycle is abundantly flowing and you have plenty of alpha-ketoglutarate, then that can readily be converted into amino acids. Okay, everybody with me? Okay. A lot of information there. Again, not asking you to regurgitate that to me. Let's look at the reactions of a couple of a few amino acids. Here's one, serine. Okay? Serine is one of the amino acids that can be made from 3-phosphoglycerate. 3-phosphoglycerate was that intermediate in glycolysis, gluconeogenesis, and in the Calvin cycle. Okay? 3-phosphoglycerate, okay, through a couple of steps, ends up as serine. We start with a 1,2,3 carbon molecule. We end up with a 1,2,3 carbon molecule. We start with a phosphate on the end. We end up with an OH on the end. We start with an OH up here. We end up with an amine over here. Again, I'm not asking you to memorize the reactions. I'm not even asking you to memorize the intermediate names, but I'm just showing you the simplicity with which a molecule like a glycolysis intermediate can be made into an amino acid that's useful for the cell. So this glycolysis pathway is not in isolation from everything else. Okay. What do I want to say here? If we look at this slide, you can see several things on here. Okay? First of all, at the very bottom, what do you see? You see something called tetrahydrofolate. I want you to remember that name. You can call it THF. And tetrahydrofolate is a molecule that's very important for the synthesis of nucleotides. It's very important for the synthesis of nucleotides. Okay? Bacteria make their own tetrahydrofolate. We don't make our own tetrahydrofolate. Okay? We get it from organisms that we eat. Now, I tell you that because one of the central things of tetrahydrofolate is this thing called PABA right here, para-aminobenzoic acid. Anybody know why PABA is important? Anybody know what PABA is used for? You do. I know you do. You've heard it before. How many think you've heard of PABA before? Nobody? One. Two. Maybe you've heard about it as PABA-free something. Does that sound like something you've heard of? How about sunscreen? PABA-free sunscreen? Check the labels out sometime. PABA is a sunscreen that you can apply that absorbs UV light and helps protect you from the sun. Some people think PABA isn't good to put on them, so they sell different sunscreens they call PABA-free. Okay? Para-aminobenzoic acid. That's a central part of this tetrahydrofolate. We have a class of drugs called sulfonamides that resemble the PABA enough that they inhibit the bacterial enzyme that makes tetrahydrofolates. Consequently, this is a useful way to kill bacteria selectively because we don't make THF. Okay? Bacteria have to make their own, so if you can inhibit the enzyme that helps them to make it, bang, you kill bacteria. Sulfonamides are useful for that purpose. Okay. Here's the use of a tetrahydrofolate. What are folates useful for? Folates are very important for what we call one-carbon metabolism. One-carbon metabolism. Here is serine. Serine has how many carbons? It's got 3, right? Here is glycine. Glycine has 2 carbons. Something's happened to one of the carbons, right? One of the carbons has been donated from serine to tetrahydrofolate to make this mouthful of a name of a molecule over here. And here's the extra carbon right there. Okay? Single carbon metabolism. A single carbon was taken from serine and donated to this folate to make this other folate. This is a folate, this is a folate. Okay? It turns out that folates can not only accept single carbons, but they can donate single carbons, and that will become very apparent and important when we talk about nucleotide synthesis, because the synthesis of nucleotides like thymidine requires donation of single carbons, and those single carbons come from folate. Okay? What do we have going on here? I show you this one because cysteine is an interesting amino acid. It actually involves sulfur metabolism. Okay? Cysteine is one of 2 amino acids that have sulfur, the other being methionine. And cysteine is the only one that has a sulfhydryl. Sulfur metabolism, we don't really get a chance to talk about in this class, but sulfur metabolism is important like nitrogen metabolism. Specialized cells have specialized reactions for absorbing and using sulfur in the environment to help us make amino acids like cysteine. Okay? There's a bunch of reactions to memorize. No, you guys know me too well. Everybody just shut up, Ahern, and get on with this damn lecture, right? Okay. So this is simply showing how an oxidized form of sulfur can be reduced ultimately to produce something that is useful for making, like in the example we saw before, cysteine. So sulfur has to be, if we have sulfur in the sulfate form, it has to be reduced to get to the sulfide form so that it can be used to make cysteine. All right. Now, most of what I've shown you so far is fairly general. I'm going to show you something specific here that's important. I told you how folates were important for what I called single carbon metabolism, right? They absorb single carbons, they donate single carbons, and they're very important for those movement of single carbons. That's one group of molecules that do single carbon metabolism. A second group of molecules that do single carbon metabolism is this guy right here, S-adenosyl methionine, or you can call it SAM, S-A-M. S-adenosyl methionine is derived from an adenosine group, which is this part over here, linked to a methionine, which is this part over here, and this carbon on the end of methionine is very, very unstable, meaning that this S-adenosyl methionine can donate that methyl group to something else. Okay? So S-adenosyl methionine is a single carbon. Specifically, it's a methyl donor. It will donate methyl groups to reactions. And it comes from combining an adenosine with a methionine. When this guy has donated its methyl group, what we're left with is called S-adenosyl homocysteine, S-A-H, S-adenosyl homocysteine. So S-adenosyl homocysteine is lacking the single carbon. S-adenosyl methionine has the single carbon. Oh, yes, question. Methionine? S-adenosyl methionine is SAM, meaning it has the methyl. That's the M part. S-adenosyl homocysteine, or SAH, lacks the methyl. Okay? Okay. Here are the requirements of amino acids for humans. It's not absolute. You'll see some of these say essential for children, but not necessarily for adults, meaning that at different stages of your life, you have greater needs or greater ability to make these and so forth, so it's not a rigid wall that's between them. But in essence, we have about 10 amino acids that are essential and about 10 amino acids that are non-essential. If you look at the non-essential ones, you'll see several I've already talked about that are products of transamination reactions. There's alanine. There's asparagine. There's aspartic acid. There's glutamic acid. There's glutamine. Okay? All of those can be made in transamination reactions. Transamination reactions are widespread. They're abundant. And consequently, if we have the precursors, like in the case of glutamic acid, we have alpha-ketoglutarate, it's simply a matter of swapping the oxygen and the nitrogen to make glutamic acid. Others are more complicated to make. And in these cases, in cases of the essential, we're lacking one or more enzymes necessary to make these amino acids. So these guys on the left have to be in our diet. Okay? Questions? All right. Now, that's the synthesis of amino acids. We need amino acids, so we have to synthesize them or we have to eat them. What happens when we eat them? What results in these guys? Okay? These amino acids can be broken down in, actually some can be broken down in more than one way, as you will see. We describe these amino acids as being either glucogenic or ketogenic, or both, glucogenic and ketogenic. Some cases, part of the molecule makes a glycolysis type intermediate. Part of the molecule makes an alpha-keto type intermediate. Okay? But suffice it to say that most of the amino acids are glucogenic, meaning that they can ultimately be made into glucose, at least part of them. Okay? Be ultimately made into glucose. Well, look at that. There's a lot of things there that we can use to make glucose. So people who are on, for example, a low-carb diet, high-protein diet are getting plenty of things they can use to make glucose. Okay? Now, we turn our attention now to dealing with all of that nitrogen that we've been sort of sloughing around and talking about. But now we see that, as I told you, nitrogen compounds like ammonia and ammonium ion, the only difference is a proton there. So if you put ammonia in water, you create ammonium hydroxide. And I'm sure in freshman chemistry lab you remember ammonium hydroxide because it has its strong smell of ammonia because ammonia is leaking out of the solution. That is very toxic. And so the body, if it has too much in the way of ammonium ion or ammonia, whatever you want to call it, can die. Okay? So the cells have to balance that, and they have to have ways of excreting or getting rid of excess ammonia. Okay? They have to have ways of dealing with that excess that they have. In us, as I noted before, we excrete excess nitrogen in the form of urea. And urea, as you can see, contains 2 amine groups and a carbon. It's a very efficient way to get rid of a fair amount of nitrogen is by forming urea, as we'll see happens in a little bit, and excreting that. Not all organisms excrete urea. Okay? Birds, for example, excrete excess nitrogen in the form of uric acid. Uric acid turns out to be a very interesting compound. And I'm going to describe it here because I think this lecture is really needing something very interesting at this point because I can tell by your reactions that it's getting to be a little long in the tooth. Okay? Uric acid is a very interesting molecule. When I talk about nucleotide metabolism on Friday, we will see that uric acid is a breakdown product of nucleotides. We make uric acid in our bodies. We don't excrete uric acid. We excrete urea. But we do make uric acid in our bodies. And uric acid is, as I said, a breakdown product of nucleotides, specifically purine nucleotides, like guanine and adenine. Okay? Uric acid, when we break it down, uric acid turns out to be not very soluble in water. That's a problem. It's not very soluble in water, and because it's not very soluble in water, it will crystallize. And when it crystallizes, it comes out of solution. So instead of being nice and dissolved in our bloodstream, it comes out as a crystal, and it turns out that when it crystallizes, it's heavier than most of the things in our body, and so it goes to the lowest point in our body. And uric acid commonly will accumulate in the big toe. Anybody know anybody who has gout? Gout is first realized by people who discover a tremendous pain in their big toe. Because what's happening is the uric acid has crystallized. It's crystallized around a nerve in the big toe and caused a tremendous amount of pain because it's stimulating that pain nerve. Okay? Now, gout used to be known as rich man's disease. Back in the Middle Ages, diets weren't what they are today, and so the only people who got gout were those who had diets that were rich in purines. These included things like red wine and red meat. Today's world, at least in the United States, people seem to have an abundance of red wine and red meat, so gout is more evenly distributed among the population. That doesn't mean because you eat red meat and drink red wine that you're going to have gout. No, it doesn't. But it means you'll be more likely to have it than someone who does not have that. There are drugs today for treating gout that are pretty good. But a very interesting component about gout, okay, A very interesting component about gout relates actually to the disease known as multiple sclerosis. There appears to be some protective effect against multiple sclerosis. People who have gout tend to have a lower incidence of multiple sclerosis. It's not understood why, okay? But there are maybe some benefits to having gout. It's thought perhaps that the uric acid acts as a kind of an antioxidant and may help prevent against oxidative damage that can result in multiple sclerosis. Another interesting thing about uric acid, birds excrete it. So the white poop that you see birds that have and so forth is a lot of that's uric acid. All right? They don't precipitate it. They don't seem to have problems with it like we do. But there is one organism that does precipitate it, that does have problems, and it's a very odd place for it to appear on the evolutionary scale. Anybody ever have a Dalmatian? Dalmatians actually excrete uric acid. They don't excrete urea. And Dalmatians, and I've never had one, so maybe those of you who've had Dalmatians can tell me this, tend to be much more sickly. They will have much more difficulty dealing with pain because they're loaded with uric acid, and it accumulates in their joints and stuff. And so that's a real problem. Who had Dalmatians? So I saw some yeses earlier. Nobody? Did you say you had Dalmatian or not? Oh, you didn't have one. Okay. Nobody's had Dalmatian. Okay. Anyway, I'm told that's the case with Dalmatians. I've never had a Dalmatian, so I don't know. I had a friend who had one. He said he was at the vet all the time with his doctor or with his dog because the dog was having a real problem. Okay. Well, I've decided my theme for the last 2 lectures, besides being nitrogen metabolism, is actually related to nutrition as well. I promised you a song about Dairy Queen that I've talked to the student who's recording, and I don't think she's going to have it ready. So we will probably sing that song on Friday. But for today's song, I have another song about nutrition that I think you will enjoy. It's recorded by some good friends of mine. It's called Your Poor Veins, and it's to the tune of an old song by Carly Simon known as You're So Vain. Okay? Let me get that for you. And All right, guys. See you Friday.